Good afternoon, everyone. Okay, so today we'll discuss about the histology of pancreas and histology of suprarenal gland, or also called as adrenal gland. Now, in histology of pancreas, already this topic we have discussed in the lecture hall last week. Okay, so today we'll see in detail. Along with that, I'll show you the slides also. Okay, so what do you remember anything what we have discussed last week in the histology of pancreas? Before starting with the pancreas, that day what I have mentioned while explaining about the liver, from now onwards what we have to follow? Four? Four basic layers will not be applicable. Then what else we have to know? Yes, stroma and parenchyma. Before that, connective tissue capsule will be there. If you remember, connective tissue capsule will be there. Okay, that connective tissue capsule is completely covering the gland. After that, what happens? The connective tissue capsule will send septa inside the gland to divide it into small lobes, or in sometimes it is also called as small lobules. Through that, what is passing? Blood vessels, nerve, lymphatics, these are entering the gland. Okay. After that, we are getting stroma and parenchyma. Stroma means the supporting part, and parenchyma means the secretory part. Okay. So in the same way how we have discussed the liver, in the similar way we have to discuss about the pancreas. Okay. But what is extra thing in case of pancreas? If you see the parenchyma of the pancreas में क्या रहता है? Secretory part of the pancreas is made up of how many components? Two components are there, exocrine component and endocrine component. Exocrine component is secreting pancreatic juice, okay, where pancreatic enzymes are present. And endocrine part, what is the function? Produce hormones. Okay. So like this you have to study about the histology of pancreas. Okay. That means when we'll discuss about the exocrine part, the cells will be different. Next, you should know that in case of exocrine pancreas, along with the pancreatic cells which are secreting the enzymes, what else we can expect? If the enzymes are secreted, how it will be carried? Duct. That means duct system are also present. So duct system is included in which part of the pancreas? Exocrine part of the pancreas. Okay. But in case of endocrine part, ducts are not there. Instead of that, what is present? Blood fenestrated capillaries are there, where hormones can be poured directly into the bloodstream. Okay. So that is the way you should study about the pancreas. As you can see, pancreas is a covered by very thin connective tissue capsule. Okay. And that will extend inwards, septa, partitioning the gland into lobules, same like the liver, whatever we have discussed. So a thin connective tissue covering is there, okay, which will send a septa, and then it will divide the pancreas into lobules. Okay. Now through that connective tissue, septa, blood vessels, nerves, lymphatics, they are entering the gland. Okay. So that part will constitute the stroma. That part will constitute the stroma of the gland. Okay. Next, come to the parenchyma of the gland. Parenchyma of the gland, that means the secretory part. So just now we have discussed that secretory part or the parenchyma is divided into two different elements. Okay. So parenchyma of the pancreas is divided into pancreatic acini and pancreatic islets. So what is pancreatic acini and what is pancreatic islets? Acini means the exocrine part. Pancreatic islets means the endocrine part. Okay. Along with that, the smooth connective tissue is also present inside the gland. Okay, that is the stroma of the gland. Next, the interlobular septa house the interlobular duct, blood vessel, nerve, okay, lamellar corpuscles, okay, which are special type of sensory receptors. So these are present in the stroma or the supporting part. Okay. So like this, you have to segregate what is there in the stroma of the gland and what is there in the parenchyma of the gland. Okay. In stroma, what we are getting? First of all, the connective tissue capsule, then a septa, along with that blood vessels, nerves, okay, and some important sensory receptors are also present. Another important point which is extending through the septa, that is the interlobular ducts. Okay, interlobular ducts are also traveling. So this will come under the supporting part. Okay, what about the parenchyma? As I said, parenchyma में दो part रहता है, that is the pancreatic acini, that is the exocrine part, and pancreatic islets, that is the endocrine part. Okay, now we'll see one by one. So already you know that pancreas is made up of both exocrine and endocrine part. So if you see the exocrine part, which is secreting the enzymes, pancreatic enzymes, okay. So the glands which are present, now unless and until glands are there, from where the secretion will come, right? So they are made up of compound tubulo-alveolar serous gland, the exocrine part of the pancreas, okay. And the bulk of the pancreas is made up of exocrine part only, okay. Bulk, approximately 98% of the pancreas is made up of exocrine part. Just imagine 98%, and only 2% is made up of endocrine part. Okay, so that's why bulk is made up of exocrine part. That means mostly you will see the glands, this compound tubulo-alveolar glands. Now the acini are present, which synthesize and secrete variety of enzymes, which is essential for the digestion. Okay, now the exocrine component, as I said, okay, it is in the bulk, which consists of many serous pancreatic acini cells. Okay, many serous pancreatic acini are present. So acini means multiple, acinus means single. Okay, that means a gland is there, okay, and inside the gland a small lumen is there. That particular structure is called as acinus. Okay, suppose a gland hai, one gland, inside the gland a narrow lumen is there, okay, or you can say a small cavity is there. So जो secretion हो रहा है, that is coming into that lumen, or the coming into that cavity, and by the duct it is thrown out of the gland. So that particular type of structure is called as acinus. Okay, and inside the gland you will be getting the acinar cells, which are responsible for production of the enzymes. Now this acini, what is the main function? They secrete variety of enzymes. Why and what is the importance of that enzyme? They helps in digestion. Next, the endocrine part is much smaller but equally important. It is composed of pancreatic islets, or also called as islets of Langerhans. Now, which appear as island of cells dispersed between the pancreatic acini. So in between the pancreatic acini, you will see islands of cells which are dispersed throughout the gland. Okay, majority of them, where they are placed, pancreatic islets, anyone? Yes, in the tail of the pancreas, majority of the islets are present. Okay, but in other parts also it is there. Now the islets cell produce and secrete hormones to regulate glucose, lipid and protein metabolism in our body. So this is the function of the hormones which are secreted by endocrine part of the pancreas. Okay, now as I said, in order to study the parenchyma of the pancreas, what we have to do? We have to segregate it. First we will be discussing about the exocrine part of the pancreas, followed by the endocrine part of the pancreas. Okay, so exocrine part में क्या रहता है? Just now I have mentioned acini are there. Okay, pancreatic acini. Okay, and inside the acini what is present? Acinar cells are present. Okay, and those cells which are present inside the acini, they will produce the enzymes which are required for digestion. Okay, so exocrine pancreas, they are made up of branched acinar gland and surrounded incompletely by lobulated and delicated loose connective tissue. Okay, that means if you see the pancreatic acini, okay, so multiple acini, if you take one particular lobule, as I said, the connective tissue septa will divide into many lobules. If you take one particular lobule, at that time what you will see mostly they are made up of acini and few islets will be present. Now this particular acini, these are branched acinar gland and surrounded completely by lobulated, delicate, loose connective tissue. Okay, so this loose connective tissue will be encircling the particular pancreatic acini which are present inside the lobule. Okay. Next, if you see the epithelial lining, now what is the meaning of this epithelial lining? Here I have said no mucosa, submucosa. So epithelial lining का मतलब क्या, meaning क्या है? Yes, inside the acinus. एसिनाई के अंदर क्या रहता है? एज आई सेड एसिनर सेल्स आर प्रेजेंट. एसिनर सेल का फंक्शन क्या है? to produce the enzymes required for digestion. ठीक है, तो I am talking about those acinar cells. Okay, so those acinar cells are made up of which type of epithelium? These are simple columnar epithelium. The acinar cells which are present inside the acini, okay, they are simple columnar epithelium having narrow luminal and broad basal surface. Okay, some modifications are there, not exactly they are simple columnar epithelium. They appear as simple columnar epithelium with some modification. And what is this modification? Narrow luminal and broad basal surface. Okay, nuclei are spherical and basally placed. Next, if we use H&E staining, okay, at that time how the cells are taking stain, the acinar cell, as you can see, the secretory cell shows basal basophilia. That means basal part will be basophilic and apical part will be eosinophilic. That is the important characteristic of the acinar cells. Okay, basal part will take basophilic stain and apical part will take eosinophilic stain. Okay, when we are staining with H&E. तो इसका मतलब क्या है? What is the meaning? That means if you see the cell, the basal part will appear dark or you can say blue or purple in color, और जो ऊपर वाला part है apical part, that will appear pink or red in color, mainly pink in color. Okay, this is the appearance. Why I am telling you this? Under microscope you have to identify this. Okay, because you have to identify the acini. Okay, you have to identify the acinars. Inside the acinars you have to identify the acinar cells. How will I identify the acinar cell? Based on these particular points. Okay, basal basophilia and apical eosinophilia. Okay, next basal basophilia is because of the rough ER, whereas apical eosinophilia is because of secretory vesicles. Okay, zymosome granules and Golgi apparatus. Because of this. Okay, now another important point. If you see the upper part, apical part of the cell, okay, it is granular in appearance. Why? Because of the zymosome granules. Okay, I will show you the diagram, don't worry. And from these zymosome granules only the secretion of the enzymes are taking place. So zymosome granules, these are secretory granules, contain digestive enzymes, okay, protease, amylase, lipase, nuclease. Okay, these are the different types of enzymes which are secreted by these zymosome granules. And very important, because these enzymes only they will complete the digestion. Next, another type of cells are there. These are called as centroacinar cells. Okay, centroacinar cell. Now, what is the meaning of these centroacinar cells? Centro means they are present at the center. Of which part? Center of the acinus. So if you take one particular acinus at the center, you will see a particular type of cell which are different than the acinar cell. मतलब acinar cells का जो appearance होगा, okay, and the centroacinar cell का जो appearance है, that is completely different. And centroacinar cells are present at the center of the acinus. Now the question is, which type of cells are these and what are the functions of these centroacinar cells? Actually, centroacinar cells are nothing but the cells of the duct. Okay, तो क्या होता है, जो suppose acini is there, inside the acini, okay, you can see inside the acini like this duct is going in. So this is the invagination of the duct. Okay, suppose this is acini, this is the invagination of the duct. तो duct क्या होता है? Invaginate होता है. That means inward protrusion of the duct inside the acini. तो suppose ऐसे हुआ है, that means if you take the section over here and if you prepare the slide, then what will appear? This particular part is completely made up of acini. Acinus, that means acinar cells will be there. But at the center, you will not get the acinar cells. Instead of that, you will be getting cells of the duct. Okay, so that's why it is called a centroacinar cell. It don't have directly any secretory functions like secretion of the enzyme. But one important part, it adds bicarbonate ion. Okay, when the pancreatic enzyme is flowing through the duct, at that time bicarbonate ions are produced by this particular centroacinar cells, which are added into the pancreatic enzymes to maintain the pH. So I hope it is clear about the centroacinar cells, everyone. Now see, centroacinar cells, they are located at the center of the acini and they represent the beginning of the intercalated duct that invaginate the acini. Okay. Next, if you see the centroacinar cell, they show pale staining cytoplasm with centrally placed flattened nucleus, almost like squamous epithelium. That means if you see the appearance of the centroacinar cell, how it will appear? Squamous like appearance. Okay, centroacinar cells secrete bicarbonate ions into the pancreatic juice. Okay, now please concentrate in the diagram. So what is this? Anyone? What is this? ये पूरा structure, what is the name of this entire structure? This is one acinus. Okay, like this multiple acini are present inside one lobule of the pancreas. So from one lobule I have taken one acinus, and if you see one acinus, it appears like this. ठीक है, तो इसमें क्या ये सब क्या है? What are this? What are this? These are acinar cells. ठीक है. Now how you will identify the acinar cells? Under H&E staining, basal basophilia, apical eosinophilia. ठीक है. This is how we can identify the acinar cells. ठीक है. And can you see at the apical part granular appearance? Why? Because of the zymosome granules. From there only the enzymes are produced, enzymes are secreted. ठीक है. Next, if you see the center of the acinus, can you see some cells? Can you see some cells? Now these cells are not the part of acinus, but these are part of intercalated duct. ठीक है. These are the cells of the intercalated duct, and that duct is invaginated into the acinus. Means acinus के अंदर चला गया है. ठीक है. And after that, if you are taking the section, then at the center you will be getting this kind of cells. Okay. They are what is the appearance of those cells? They are pale staining and squamous like appearance. Okay, that's and what is the main function of these cells? Bicarbonate ion. ठीक है. तो whenever यहाँ से जो okay, so what is the function of centroacinar cell? They will secrete bicarbonate ion. So when the pancreatic enzymes they are flowing, okay, pancreatic juice through this intercalated duct, they will secrete bicarbonate ion, which will be mixed with the pancreatic juice. Okay, like this it maintains the pH of the pancreatic juice. Can anyone tell me what is the pH of pancreatic juice? It is more than 8, 8.3 or 8.5. Yeah, approximately 8.3 to 8.5. That means alkaline or acidic? Alkaline in nature. Okay, next, now this is what? This is the schematic diagram of pancreas. What you can see over here? Can you see the pancreatic acini? See, first of all, you have to see interlobular connective tissue. See here, this is one interlobular connective tissue. This is interlobular connective tissue. So like this, as I said, connective tissue capsule. From connective tissue capsule, septa is extending, which is dividing the gland into small lobules. तो ये देखो, ये एक lobule है. This is one lobule. This is one lobule. This is one lobule. Okay. Can you see the connective tissue? See, this is the interlobular connective tissue. This one. ठीक है. So this is one particular lobule, this is one particular lobule, this is one particular lobule. ठीक है. Now take one particular lobule, any one. Okay, suppose I am taking this particular lobule. What you can see in this lobule? क्या दिखाई दे रहा है? First, what you have to see? First, you have to see. Though we are discussing the exocrine part, that means you will be focusing on acini. Can you see the acini? ये देखो. These are nothing but the acini. Okay, these are acini. Now in between the acini, what is this? Islets. These are islets. No. Centroacinar is different. I will show you. Don't worry. So this is the islet. So all these are the acini. As I said, majority is made up of acini, and pale staining area which will be present. These are the islets. Okay, the endocrine part. Okay. Next, along with the acini, can you see the duct? See, this is the interlobular duct. So this interlobular duct, it is larger. Okay, if you see the smallest one, that is the intercalated duct. उसका diameter कम होगा. As you can see, this is the intercalated duct. Now this duct, always you can see, these are related to the acini. Why they are related to the acini? The ducts, why they are related to the acini? Because they have to conduct the enzyme. Acinas का function क्या है? to produce enzyme. Once the enzyme is produced, it has to be conducted. How it will be conducted? With the help of the duct. That's why the ducts are always related to the acini. Okay? Now, if we, what is this? ये क्या था? Islets. See islets. These are also islets. Okay? Can you see any duct inside the islet? No. Instead of that, what you can see? Blood capillaries are there. Fenestrated capillaries are there. Okay, like this you have to differentiate. Another important point to differentiate between acinas and the islets is islets are pale staining and acini are dark staining. Okay, this is how you can easily differentiate. Next, what you have to see? You have to see the ducts. Around the acini क्या दिखाई देगा? Ducts will be seen. Either interlobular duct or intercalated duct. Interlobular duct है तो diameter it will be more. If it is intercalated duct, diameter will be less. Okay, along with that, lining epithelium also changes. Okay, but instead, in case of islet, if you see inside of the islet, no ducts are there. Instead of that, what you can see? Fenestrated capillaries are there. Okay, next is the duct system of the pancreas. So, exocrine pancreas are having ducts, different types of ducts are there. Okay, now do you remember that day also I have mentioned that whenever the duct, the diameter of the duct changes, that means if you go from smallest duct to the largest duct, at that time what will happen? First of all, the diameter will change. Okay, diameter will keep on increasing. Along with that, what will change? Lining epithelium also changes. Most of the organ, you can say almost all the organs where those ducts are present. Okay, we have seen about liver also. Now we are discussing about pancreas. Okay, in upcoming classes, means after your second term, we'll be discussing about the salivary glands. At that time also you will see like these ducts are there. Okay, so directly you will not get the bigger duct. Smaller ducts, okay, gradually they will join together to form the larger ducts. So though two changes are taking place. First of all, from smaller duct to larger duct, diameter will change. And second point is lining epithelium also changes. Okay, lining epithelium also changes as you go from smaller duct to the larger duct. So same thing, same phenomena will be taking place over here. So in case of ducts of the pancreas, first the smallest one which we are getting, that is the intercalated duct. From intercalated duct, what we are getting? Interlobular duct, and from interlobular duct we are getting the main and accessory pancreatic duct. Okay. Now if you see the lining epithelium, the interlobular ducts, okay, these are surrounded by connective tissue where the septa is there, then autonomic nerve fibers, smooth muscle fibers, all those things are present, which helps in conducting the enzyme through the duct. If you see the intercalated duct, okay, this where they begins as the centroacinar cells, and they are lined by simple squamous to very low simple cuboidal epithelium. Okay, that is the intercalated duct. Next, intercalated duct will open into interlobular duct. They are lined by cuboidal or low columnar epithelium. Okay, see the lining epithelium is changing. Next, if you see the main and accessory pancreatic duct, they are lined by simple columnar epithelium. Okay, this is how the lining epithelium changes. So in most of the cases I have seen, जो smallest duct होता है, in that lining epithelium is almost squamous or low cuboidal. Okay, after that low columnar, and then finally in case of larger ducts it is simple columnar epithelium. So in case of the exocrine part, the ducts which are there, the main function of those ducts are add bicarbonates and water in pancreatic secretions. Next, exocrine pancreatic secretions are under control of secretin and cholecystokinin hormones as well as autonomic nerves. That means जो secretion होता है, this thing already you have studied in physiology. I am not going much in detail about this. Okay, जो secretion होता है exocrine pancreas का, okay, it is under control. Now it is under hormonal control as well as it is under autonomic control. Okay, for hormonal control, cholecystokinin hormone and secretin hormone, these are the main two hormones which are controlling the secretion, and along with that autonomic nerves are also there. Now here you can see the, so what is this? Anyone? Acinus, one particular acinus. After that, can you see the acinar cells? Can you see the acinar cells? Yes, see, you can see the acinar cells. After that, what are these dot things? Zymosome granules. Very good. After that ये बीच में क्या है? What is this? Which cell is this? Centroacinar cell. Centroacinar cell is a part of intercalated duct which is invaginated inside the acinus. So you can see the duct is going inside the acinus. Okay, that means after that if you take the section, so the cells which will appear at the center, these are called as centroacinar cell. Function क्या है? Bicarbonate ions. They will add bicarbonate ions where? In the pancreatic secretion, pancreatic juice, to maintain the pH. Okay, after that here you can see the intercalated duct. Okay, after that you can see the interlobular duct. Now in the entire exocrine pancreas, what are the different types of cells present that you can see? Okay, so in case of acini you can see simple columnar cells. Okay, basal, basophilia, apical, eosinophilia. Next, centroacinar cell, that is simple squamous to low cuboidal epithelium. Then intercalated low cuboidal epithelium, interlobular, that is cuboidal to low columnar cell. Okay, and main and accessory pancreatic duct, simple columnar epithelium. Okay, now one concept is there about the striated duct. Striated duct pancreas में नहीं रहता, it is absent, but it is present in case of salivary glands. So after second internal in head and neck phase, we have to study about the salivary glands. Okay, at that time we will discuss what are these non-striated duct. Next, another important type of cells which are present, okay, in the pancreas, these are called as pancreatic stellate cells. Okay, pancreatic stellate cell. Have you studied this one in biochemistry? Not yet. Okay. तो pancreatic stellate cell जो होता है, predominantly they are located in the pre-acinar, preductal and prevascular spaces of the pancreas. These are the areas where this pancreatic stellate cells are present. Okay, stellate cell liver में भी रहता है. Last time we have discussed. Now what is the main function? This will maintain the normal tissue architecture. Pancreas का जो normal tissue architecture है, that is maintained by this particular type of cells. Okay, if you see this particular cells, the main function is they will synthesize the extracellular matrix protein matrix degrading enzymes. Okay, tissue inhibitors of metalloproteinases. These are secreted by the by these particular cells. Okay, like this it is maintaining the tissue architecture. Now there are some cases, for example pro-inflammatory cytokines, when it increases oxidative stress, hypoxia, hyperglycemia, increased intestinal pressure. Okay, they can activate the cell. Now during that particular moment what happens, various growth factors, activated PCS can secrete various growth factor. Okay, and those growth factor will cause the proliferation of pancreatic cancer. Okay, pancreatic cancer cells inhibit their apoptosis and enhance their invasion ability. That means it can invade to the other tissue, other organs. So pancreatic stellate cell, once it is very important, okay, because it is maintaining the normal architecture, but if it is get activated, then it can cause proliferation of the pancreatic cancer. Now here you can see pancreatic stellate cell. Now in the previous diagram I have not shown you the pancreatic stellate cell. Okay, do you think in under H&E staining can you see the pancreatic stellate cell? Stellate cell means star shaped होता है. Okay, you can see these are the pancreatic stellate cells. ये जो green color में दिया है, these are the pancreatic stellate cells. Under H&E staining you can't see the pancreatic stellate cell. Okay, for that special type of stains are required. So in order to stain the pancreatic stellate cell, okay, in order to stain it, some biomarkers are there. Okay, so with the help of those biomarkers, the staining of the cells can be done. Okay, now in those biomarker, desmin is there. Okay, then alpha SMA is there. Like this, different types of biomarkers are there. Okay, so in order to stain that cell, particular those biomarkers are used. Okay, to see the cells, the position of the cells. So the pancreatic juice which is there, what is the amount of pancreatic juice secreted in our body in an adult? 500 to 800 milliliter per day. And if you see the pH, it is highly alkaline, 8 to 8.5. Okay, तो जो pancreatic juice secrete होता है, in that what is present, only pancreatic enzymes are not there. Okay, along with that other things are also present. Okay, as you can see. So water 99.5%, solid 0.5%. Okay. After that you can see organic substances are there and inorganic substances are also there. Mostly in case of organic substances we are getting the enzymes. See the list of enzymes. Okay. So proteolytic enzymes are there, lipolytic enzymes are there, and amylolytic enzymes are there. Okay. I know this thing you have studied in physiology. Next, here you can see the list of enzymes secreted from the pancreatic acinus. After that activator, then act on which particular substance, and then what is the end product. Okay, so that is all about the exocrine part of the pancreas. Next is the endocrine part of the pancreas. So only 2% of the pancreatic tissue is made up of pancreatic endocrine part of the pancreas. Remaining 98% is made up of exocrine part. Okay, so endocrine part में क्या रहता है? What is present in the endocrine part? Islets or islets of Langerhans. Majority of them where they are situated at the tail of the pancreas. Okay, now if you see the islets, so they are dispersed throughout the exocrine component of the pancreas, but mostly they are located at the tail region. Means their population is more at tail region. Now how to demarcate the islets? How will differentiate between the acinus and the islet? Yes, islets are pale staining. Okay, and if you see the acinus, they are dark staining. Another important point is there, that is, if you see the islets are demarcated from rest of the parenchyma by delicate sheath of reticular fibers. Okay, so जो islets रहता है, where cells are present, around that what you can see, delicate sheath of reticular fibers are present. So clusters of cells will be there. Around those clusters of cells, what are present? Delicate sheath of reticular fibers are present. Okay. Next, pancreatic islets are spherical clusters of polygonal endocrine cells. Okay. On histological slide H&E, they are large pale staining cells enveloped intensely staining basophilic pancreatic acini. Means around that darkly staining basophilic acini will be there, and at the middle or at the center you will be getting pale staining area. Okay. So those pale staining area, what you can see, clusters of spherical polygonal cells. This is the arrangement, or you can say orientation, of the pancreatic islets. Now the cells of the islets are connected to each other with the help of desmosome. Okay, now they are not widely separated. The cells, one particular cell, another cell, they are attached to each other. How they are attached? With the help of desmosomes and gap junctions. Like this, they are forming cords of cells. Next, pancreatic islets are permeated by many fenestrated capillaries. That means in between the cells you can see fenestrated capillaries. Very important, because through those fenestrated capillaries only the hormones are drained. Next, if you use H&E stain, okay, you can identify, yes, this is pancreatic islet, this is acinus, but you cannot differentiate the different types of cells which are present inside the islets. Okay. For that, what you have to do? Special type of stain are used. That is called Melroy-Azan staining. This is a special type of stain, and for that a special type of fixing method is there to fix the cells. That is called as Zenker's formal fixation. If you do that, what you can see, 3 different types of cells can be seen. That also in 3 different color. If you see red color, that is for alpha cells, brownish, that is for beta cells, and blue color, that is for delta cell. Okay, that is Melroy-Azan staining. If you use that, then you can see these 3 different colors for these 3 different types of cells for islets. Next, another type of staining method can be used, that is immunohistochemical staining method. If you use this particular type of method, along with that you can also see different types of cells. For example, F cells are also called PP cells, then D1 cells and enterochromaffin cell. Okay, with the help of this particular type, other type of cells can also be identified. Now in the endocrine pancreas, what are the different types of cells present? So first of all, alpha cells are there. What is the function? Glucagon. If you see the population of the alpha cells, it is approximately 15 to 20% of the total cell population of islet. I am talking about one islet. Next, beta cells. Okay, what is the function? Insulin. Approximately 60 to 70% of the cell population is made up of beta cells. Next, delta cell. इसका function क्या है? To produce somatostatin. Okay, what is the cell population? Approximately 5 to 10%. Next, other cells are there, but their populations are very less. Okay, for example, PP cells are there, which will produce pancreatic polypeptide, D1 cell, vasoactive intestinal peptide. This is enterochromatin cells. They will secrete serotonin, motilin, and substance P. Okay, these are the different hormones which are secreted by enterochromatin cells. Okay, now here you can see this is a typical structure of islet. Okay, now in this particular diagram, what you can see? Can you see the cells? Different types of cells. You can see different types of cells. Now those cells are not widely separated. They are attached to each other mainly with help of gap junction and desmosome. Next, inside the islet, I hope you can see the capillaries. So inside the islet you can see the blood capillaries. Okay, and around the islet what is present? A sheath of or a covering of reticular fibers. Okay, yes sir, reticular fibers. Okay, so here you can see this is the appearance of the islets and this is the appearance of the pancreatic acinus. Now if you see the innervation of the pancreatic islet, why this is important? Okay, because the control, the way hormones are secreted, it must be controlled. Okay, that's why the innervation is very important. Now it is extensively done by the parasympathetic system. The fine branches of the parasympathetic system, they will ramify among the cells to form the plexus around the islets. Okay, so first of all they will form plexus around the islets. Now this particular fibers, they will synapse with the acinar cells before innervating the islets. So first it will be synapsing, it will be supplying the acinar cells. After that it will supply the cells of the islets. Okay, now many fibers enter the islets with the arterioles. The parasympathetic ganglia which is there, okay, situated in the connective tissue within the blood vessels and frequently associated with islet cells. Now together this connective tissue with the parasympathetic nerves, they will form neuroinsular complex. Okay, so the neuroinsular complex is formed before the nerves are supplying the pancreatic islets. Okay, let me show you the diagram. Here देखो, this is the neuroinsular complex. See, this is the neuroinsular complex. Now here you can see this is the ganglion. So this is one ganglia, this is one ganglia, this is one ganglia. This particular part is the preganglionic fiber. This is preganglionic fiber, these are preganglionic fiber. Okay, now this will come and rely over here on this particular ganglia. After that, the fibers which are coming out from the ganglia, these are called as postganglionic fibers. Okay, so this is one part. Along with that, we are also getting the fenestrated blood capillaries. Okay, and along with that, the connective tissue covering is also present. So together they are forming the neuroinsular complex. Okay, so this particular part is called as neuroinsular complex. Why this is important? Because this particular innervation will control the production of the hormone. Okay, now along with that, the hormonal control is also there. Entirely it is not controlled by the nervous system. Okay, it is also controlled by the hormones which are secreted from pituitary gland. Okay, so when you will study the pituitary gland, at that time you will come to know how the regulation is taking place. Okay, so this is a very beautiful 3D diagram where you can see this is the pancreatic islet where different types of cells are there. You can see the cell junction also. Okay, after that you can see the neuroinsular complex also. Okay. Next, this is the histology of pancreas under low magnification. Okay, so once you will put the slide under microscope under low magnification, this will be the appearance. So what exactly you can see? Under low magnification, what you can see over here? क्या दिखाई दे रहा है? तो can you see the pancreatic acini? Okay. Next, can you identify the islets? How you can identify it is pale staining. ये देखो, these are the pancreatic islets. This is one islet. Okay, then see this. These are the islets. These are the islets. Okay, but here it is very difficult to identify the ducts. ये देखो, these are the ducts, but it is very difficult to identify. Okay, for that what you have to do? Higher magnification. Okay, for that you have to go for higher magnification. Now here you can see this is the histology of pancreas under higher magnification. Okay, so here you can see these are the pancreatic acini. Okay, these are the pancreatic acini, and here you can see this is the islets. Now inside the islets, I hope you can see these are the fenestrated capillaries. ये देखो, these are the capillaries. Okay. Next, if you focus on any of the pancreatic acinas, at the center you can see the centroacinar cells also. You can see the centroacinar cells. Then you can see the zymosome granules. Okay. If you see the basal part of the cell, you can see it is purple in color. That is basal basophilia. Apical part is eosinophilia. This is the connective tissue septa. This is the connective tissue septa. This is the connective tissue septa. Okay, which is separating the lobules of the pancreas. Okay, so all those things you can see under high magnification. So that is all about the pancreas. Zymosome granules you can see. Apical part, granular appearance of it. In case of histology of adrenal gland, okay, the same way we have discussed this particular pancreas, in the similar way we have to discuss this. Okay. Now here also you can see the gland is surrounded by a thin connective tissue capsule. Okay, then it is sending septa inside the gland. Same story is followed. Okay. Now next, here one difference is, if you see the gland, okay, the suprarenal gland, mostly it is made up of two parts. That is, here we are not getting directly stroma and parenchyma. I am not telling the stroma parenchyma nahi hai, it is there. But here what we have to mention, what we have to know, that is the cortex and medulla. Okay. Now do you know the concept of cortex and medulla? Cortex means the outer part, medulla means the inner part. Okay. So here if you take the gland and if you take an LS of the gland, okay, suppose you have taken this particular, imagine you have taken an LS of this particular suprarenal gland. Okay. Once you take the LS, you can open it. What you can see inside? You can see two things. That is the cortex and the medulla. If you remember, I hope you have seen the specimen of kidney. Have you studied the coronal section of kidney? ऐसे kidney को काट के ऐसे open करके? Not yet. Okay. So then I think in the next class you will see. When we discuss the kidney, in order to study the internal structure, देखो kidney में two things are there. Internal structure is there. So in case of kidney, internal structure and microscopic structure of kidney both are different. Okay. Tomorrow I think I am having one class, so tomorrow I'll show you the internal structure and the microscopic structure. Internal structure means what you have to do. You have to take a coronal section of the kidney. If you remember the different planes which you have studied, sagittal plane, coronal plane, axial plane. Okay. So suppose you have taken kidney in your hand and you have to cut the kidney. How you will cut? In coronal section. On coronal section you have cut the kidney, then you have opened it. Okay. Whatever you can see, there also you will be able to see cortex and medulla. Okay. Similarly in suprarenal gland also, suppose you have taken the suprarenal gland, you have taken a LS. After that अंदर क्या दिखाई देगा? You will be able to see cortex and medulla. Okay. Now in case of suprarenal gland, if you see the cortex and medulla, cortex is occupying 90% of the gland. Medulla is occupying only 10%. That means medulla is smaller, thinner as compared to the cortex. Okay. Next, most of the cells, adrenal gland is a which type of gland? Any idea? And endocrine gland जो होता है, which type of gland it is or which type of organ it is? Purely endocrine. Purely endocrine gland. Okay, because as you will see, we will not discuss about any ducts which are present inside the gland, क्योंकि duct होता ही नहीं है. It is purely endocrine gland. Okay, though you are studying the kidney, you will study about the endocrine gland also. Okay, in gross anatomy. Okay, that also I mentioned. If you see the shape, both are not same. It is different. Okay, so you have to know about the shape. You have to know about the size, location, then structure. In structure, if you take the LS, what you can see. Okay, after that the connective tissue covering, then nerve supply, blood supply. Okay, all those things you have to study in gross. So now we are not going into that, it will take time. Okay, so as I said, if you take the LS, what you can see, it is made up of two things. What are they? Cortex and medulla. Okay, cortex is large, approximately 90%, and medulla is very small, approximately 10% is occupied by the medulla. Okay, now if you see the cortex, so first we'll discuss about the cortex. If you see the cortex, it is made up of cells arranged in cords. In between the cells, sinusoids are present in between the cords. Okay, see here I have not mentioned about the ducts. Ducts nahi hai. Instead of duct, what are there? Sinusoids are there, because it is completely endocrine gland. So jitna bhi secretion hota hai, all are hormones. So directly they come into the blood. Okay, now if you can see this particular diagram. Okay, see if one LS is taken, after that you can see this small particular part. Okay, is the medulla, and this entire larger part is the cortex. So cortex outer part, larger part is the cortex. Okay, the inner smaller part is the medulla. So first you will be getting one capsule, connective tissue capsule. Okay, after that you can see this is the cortex. This entire part is the cortex. A cortex is again divisible into three different zones. जो cortex होता है, that is divisible into three different zones. ठीक है, and followed by the medulla, very thin small medulla. Next, on the basis of the arrangement of the cells, the cortex can be divided into three layers, or you can see three zones. Okay, how it is divided? Based on the arrangement of the cells. ठीक है, cortex में different types of cells रहता है. What are the functions of those cells? To produce hormones, different types of hormones. Now, how they are arranged, depending on them, the zones are divided. Okay, three zones are there, as you can see: zona glomerulosa, fasciculata, and reticularis. Okay, so if you see the glomerulosa, this is the outermost. Next, fasciculata, this is the middle one, which is the largest zone, and the inner one, which is smallest, that is the reticularis. Okay, these are the three different zones. Now, if you see the cells of the adrenal cortex, they have typical structure of steroid secreting cells. Okay, different types of steroid hormones are secreted. Okay, by these particular cells. Now, they do not store the secretory product, but they secrete whenever it is required. Okay, means cell will not store anything. Whenever it is required, directly it will secrete into the blood. That is the speciality of the cells which are present in the cortex of suprarenal gland. So which is the outermost layer? Zona glomerulosa. Okay, now in case of zona glomerulosa, it is constituting the 1/5 of the cortex. 1/5 of the cortex is made up of zona glomerulosa. It is the outermost layer. If you see the arrangement of the cells, how they are arranged: inverted U-shaped cords or round like acinous groups surrounded by capillaries. This is the arrangement. Okay, inverted U shape. I hope you know U. उसको उल्टा कर दो. So that is the inverted U. What? এখন না, wait, sit down. Okay, inverted U shaped or round like acinus, and in between the cells, what is present? You can see surrounded by capillaries. Now, if you see under light microscope, okay, the cells of the zona glomerulosa are polyhedral or columnar with basophilic cytoplasm and deeply stained nuclei. That means if you see the shape of the cell, particularly in zona glomerulosa, what is the shape? It is polyhedral or columnar in shape. Okay, inverted U shaped रहता है. What are the shape? Polyhedral or columnar cells. Next, what is the important function? They produce mineralocorticoid hormone, that is the aldosterone or deoxycorticosterone. Now, what is the function? This particular hormone maintains the electrolyte and water balance of the body. Okay. Next is the zona fasciculata, that is the middle layer, which is the largest one, which is occupying middle 3/5 of the cortex. Middle 3/5 of the cortex is occupied by zona fasciculata. Okay. Now, if you see the zona fasciculata, how the cells are arranged in the form of straight columns. Okay. In the form of straight columns, that also with two cell thicks. That means two layers of cell will form a straight column, and in between the column the spaces are there, and that is occupied by sinusoids. Next, with light microscope, the cells of zona fasciculata, if you see, it is polyhedral with basophilic cytoplasm and vesicular nuclei. Okay, and rich in lipid droplets. So at the apical part of the cell, lipid droplets are present. Okay. Now, these particular cells, during processing, the lipid droplets are washed away. They appear vacuolated. That's why these cells are also called as spongiocytes. Okay, and they can also store considerable amount of vitamin C. Now, what is the function? They produce a glucocorticoid, that is called cortisone and cortisol. Okay, इसका function क्या है? Widespread effect, including those on carbohydrate metabolism and protein metabolism. Okay, carbohydrate and protein metabolism is controlled by these particular hormones, and they also have anti-inflammatory effect in the body. Lastly, the zona reticularis, that is the innermost layer. Okay, the smallest one. About 5% of the cortex is covered by the zona reticularis. Okay, if you see the zona reticularis, how the cells are arranged, they are arranged in the form of reticulum or network. Okay, and these cords are separated by fenestrated capillaries. Now, if you see the cells, these are smaller than fasciculata. Now, they are darkly stained nuclei, well developed ER, and few rough ER also present. Okay. Now, these particular cells, they are having large yellow-brown lipofuscin pigment granules. These are peculiarity for this particular type of cells. Okay, now what is the function? They secrete weak androgen, okay, and small quantity of glucocorticoids. Now, here is the diagram. So, this is the schematic diagram which you have to draw in the practical notebook. Okay. So, first of all, can you see the capsule? Connective tissue capsule is there. Okay. After that, this entire part is the cortex, and this small part is the medulla. So, first, how you will differentiate between cortex and medulla? Medulla I have not discussed, but by looking at this particular diagram, how you will differentiate cortex and medulla? Medulla pale stain hota hai. Okay, cortex darkly stain hota hai. Next, in case of medulla, the capillaries which are present, they are wide capillaries. Okay, the capillaries which are present in the medulla, they are larger as compared to the cortex. Okay. Next, here you can see three different zones. So, see, this is the zona glomerulosa. See, this is the zona fasciculata, and this is the zona reticularis, and this part is the medulla. Now, how you will differentiate these three zones? कैसे differentiate? How you will differentiate these three zones? Depending on the arrangement of the cells. Depending on the arrangement of the cells. As you can see, in case of zona glomerulosa, inverted U-shaped clusters of cells are there. Okay? Next, in case of zona fasciculata, what happens? Columns of cells are there, separated by fenestrated capillaries. And in case of zona reticularis, reticulum or network-like cells are present. Okay, and in between them, you can see the capillaries. Okay, this is the arrangement of the different cells present in the different zones of cortex. And along with that, we have also discussed what are the different types of hormones secreted by them. Okay, lastly, the medulla. Okay, so if you see the medulla, structurally, functionally, it is different from the cortex, and embryologically, it is also different. Okay, that means the source, how it is derived, that is also different from the cortex. Now if you see the function of medulla, it is modified sympathetic ganglion with postganglionic sympathetic cells. Okay, now it has chromaffin cells and sympathetic ganglion. These are the two types of cells which are present. Okay, what are they? Chromaffin cells are there and sympathetic ganglion cells are there. Okay, now they are derived from the neural crest cells. Okay, now these chromaffin cells are columnar or polyhedral. If you see the shape of the chromaffin cell, okay, they are columnar or polyhedral, and basophilic cytoplasm is there. Now the cells groups or columns are separated by white sinusoids. Now why they are called as chromaffin cell? Because a special type of dye is there which is taken up by these particular cells, and this is called as potassium dichromate. Okay, so if you want to stain and identify the cells of the medulla, at that time a special type of stain you have to use, that is called as potassium dichromate. Okay, while using that particular type of dye, the cells, chromaffin cells, they will appear brown in color, brownish color appearance hota hai. That's why they are called as chromaffin cells, because they are taking this particular dye. Okay, now what is the function of this particular chromaffin cells? So they secrete noradrenaline and adrenaline into the blood. Okay, I hope you know what is the function of adrenaline and noradrenaline. Okay, now here you can see different parts. First of all, capsule is there, zona glomerulosa. Okay, you can see the zona fasciculata, and sinusoids can also be seen. You can see the zona reticularis, then sinusoids are also seen, and this particular part is medulla. Now in the medulla you can see the secretory cells. Okay, secretory cells are nothing but the chromaffin cells. The chromaffin cells are columnar shaped. Like this it can be identified. And if you see the ganglion cells, okay, postganglionic cells, they are mainly oval or polygonal shape. Next, those ganglion cell, if you see, they are having prominent nucleus. Okay, so this is how the differences are there in the cells of the medulla. ठीक है, cells of the medulla में only दो type का cells रहता है. जो medulla है उसमें दो type का cells रहता है. What are they? Chromaffin cells and postganglionic cells. ठीक है, postganglionic cell का main function है जो न्यूरोट्रांसमीटर है उसको ट्रांसमिट करना, so that it can control the secretion of the cortex. Okay, and जो chromaffin cell है, what is the main function? As I said, adrenaline and noradrenaline into the blood. Okay, now lastly this is the list. Okay, as you can see, gland region of the cell. So first region of the gland, adrenal cortex, zona glomerulosa, fasciculata and reticularis. You can see the hormones, mineralocorticoid, glucocorticoid and androgens. Okay. Now here you can see the regulation and secretion, and here you can see these are the function. Okay, so this is very important. I will request everyone to include this chart in your practical notebook while writing about the suprarenal gland. Okay, so that is all about the suprarenal gland. Now we'll go to the lab and we'll see the slides.